Good afternoon, everyone. I'm just going to give a couple minutes for people to keep joining. All right, I think people have started. Numbers have started staying steady, so we can get started. All right, so good afternoon, everyone. My name is Julia Bachman, and I'm a program manager at NINDS, and I work with the NIH's HEAL initiative. And for today's webinar, we'll be discussing RFA NS 25-023 for studies to enable analgesic discovery. I'm also joined by my colleague, Dr. Rachel Weinberg, who'll be helping orchestrate today's webinar, in particular running the slides. Next slide, please. Okay, so just some logistics. The bulk of today's presentation will be providing you with an overview of the funding opportunity, its goals, scope of activities, and general points to consider. So that'll take approximately 30 minutes. And then we'll open it up to answer any questions that you may have. There is a Q&A box at the bottom of your screen, so please use that box to post all questions. Otherwise, attendees should be muted. That stated, if you have project-specific questions, it would actually be better to reach out via email, and then we can set up a time to discuss your project further if needed, as so often the devil can often be in the details. And then finally, this webinar will be recorded, and the recording and the slides will be made available for later reference. As I go through, you'll notice that some of the slides contain some links, so just know you'll have access to those later. Next slide. Okay, so by now, most of you are probably familiar with the NIH HEAL initiative, or Helping to End Addiction Long-Term initiative, the goal of which is to combat the current opioid epidemic through two main areas of research, one for improving treatment for opioid misuse and addiction, and the other focused on understanding, managing, and treating pain. And included within that priority area to enhance pain management is to accelerate the discovery and preclinical development of non-addictive pain treatments and advance those treatments into the clinical pipeline. Next slide. The NIH has developed a suite of programs and funding opportunities to support translational research through phase 1 first-in-human testing and beyond. So shown on this slide is just a subset of HEAL programs and opportunities that are specifically geared towards development of therapeutic interventions. And these cover multiple therapeutic modalities from small molecules and biologics to devices as well as biomarkers. Now, the opportunity being discussed today is this one outlined in the middle, RFA NS 25-023, which is an early stage translational program specifically for small molecules and biologics that is meant as a feeder program for the Pain Therapeutics Development Program, which is also listed here as RFA NS 24-019. I should also note that NS 25023 is a reissue of a recent NOFO that's NS 25012. So this was to account for the changes across NIH for the simplified review framework. Otherwise, it's essentially the same RFA. Next slide. Okay, just a very quick snapshot of the RFA. Again, it's studies to enable analgesic discovery, or the SEED program. And in a nutshell, the goal of the RFA is to support initial translational efforts that will support and enable a drug discovery program and advanced projects to the point where they can meet the entry criteria for the Pain Therapeutics Development Program, or PDDP. It's an R61, R3 milestone-driven phased award mechanism, which I'll come back to later in today's presentation. It's up to 3 years of funding and up to $350K direct cost per year. It supports small molecules, biologics, and natural products. And very broadly speaking, the scope of activities responsive to this RFA will include assay development and validation, screening for initial hit identification, and then in vitro characterization of hits and potential therapeutic agents. Finally, for groups that already have potential therapeutic agents or tool compounds in hand, you can conduct preliminary PK, PD, and efficacy studies. And then the next receipt date for this is January 28th, followed by dates in May and September, with additional receipt dates for future years listed in the funding opportunity. Next slide. Okay. Now, since the R61, R33 is essentially meant as a feeder program to PDDP, I do want to start off by taking a couple minutes to briefly touch on this next stage of funding, the goal of which is to support preclinical optimization through early phase 1 testing. Now, the entry criteria for this program are to have a promising small molecule or biologic starting point for optimization, scientifically sound assays to optimize and test the agent, and then a rigorously supported biological rationale for the intended approach. And the scope of activities covers the entire early therapeutic development process, and this includes exploratory hit to lead activities, lead optimization, selection, and characterization. It also supports optimization of pharmacodynamic biomarkers and PK/PD studies. Projects would then go through IND enabling studies, manufacturing, and finally a first in human clinical trial. And one of the unique aspects of this program is the additional expertise and resources that are provided to really facilitate drug discovery and development for academic and small business settings. So funded investigators work together with the NIH to build a lead development team where they have access to consultants with extensive biopharma experience. Projects can also utilize our CROs for a variety of activities, including medicinal chemistry, manufacturing and formulation, DMPK, GLP tox, and then also phase 1 clinical testing. So again, the R61, R33 is meant as a feeder program to get groups ready for PDDP. So I would strongly encourage you to take a closer look at this NOFO and program. So Dr. Pellimander and her team also hold several applicant webinars a year, and past ones are up on their website. I think they did one just last week. So if you want to get a more in-depth view of the program, I would encourage you to do that. Next slide. Okay, getting back to the SEED program, let's start to unpack this a bit. So the first thing I want to touch on is the mechanism, especially for those of you who may be more familiar with the standard R01 or R21 type mechanisms. This is going to be very different. So this NOFO uses the R61, R33 phase innovation award mechanism. So you have an R61 phase, a go no-go decision point, and then potentially the R33 phase. The total award time is up to 3 years with no more than 2 years in either phase. Advantages of using this mechanism are twofold. For the applicants, it allows for dependent aims, which are highly relevant for translational research and drug discovery, but often don't go over well in standard study sections. And then for the NIH, it provides a means of risk mitigation, so we can potentially take on riskier projects. Knowing the R33 phase is not guaranteed, rather it's dependent on that go-no-go decision point between the two phases that will be based in part on meeting one set of project milestones. The transition is done via administrative review, which means that review is done internally by NIH program and grants management staff. It will still require some paperwork and an application that's probably more than an RPPR, but less than a full competitive application. Next slide. The entry criteria for this program are centered on novelty, biological rationale, and relevance for therapy development. So this program is intended to support projects with some degree of novelty, whether that be around the target, mechanisms, methods, or therapeutic modality, as well as a strong biological rationale supporting a druggable target, as well as the intended approach. And this rationale should be based on rigorous and robust supporting data, whether that be preliminary data from the lab or literature-based evidence. And then projects should ultimately aim to discover novel therapeutic agents that could be advanced towards development of effective pain management strategies that are significant improvements over what currently exists. So I do want to note that for those of you who may be familiar with previous HEAL, R61, and U19 funding opportunities that were available that covered this space, one big difference for this R61, R33 is that you really need to have a strong data package supporting the target. So the bar for target validation is going to be higher here than those prior opportunities. That being said, I do want to note that the target doesn't necessarily need to be a specific molecule. So it can be a cell type or circuit, and some phenotypic screening is allowed. So just know that each of these approaches to your quote unquote target is going to have its own caveats. Next slide. All right, so let's get into the scope of activities. The next two slides include a list of the types of activities that can be supported by this RFA, although it's not meant as an exhaustive list, and there are a lot more examples that are included in the funding opportunity description. Now, this initiative is primarily focused on assay development and validation, screening for identification of initial hits, and in vitro characterization of potential therapeutic agents. So in terms of development and validation of assays, they can be assays to evaluate potency, specificity, selectivity, that type of thing, binding assays, assays for biophysical characterization, or assays to evaluate cellular uptake or target engagement. You could also have optimization of assays for medium or high throughput screening. It also supports the screening activities themselves to identify initial hits, and this can include application of biophysical and computational methods, as well as some phenotypic screening. Synthesis and in vitro characterization of screening hits or a focused set of therapeutic agents, development of bioanalytical assays or detection methods to accurately quantify therapeutic agents that might be needed for PK, as well as some limited in vitro or ex vivo biology studies for validation of an agent's mechanism of action. Next slide. Now, moving on to in vivo work, it's important to note that applications including in vivo studies must have a potential therapeutic agent or tool compound in hand with sufficient evidence to support its use for the proposed studies. So this essentially means that whatever you want to test in vivo should be outlined in the application with sufficient detail such that reviewers can determine whether those studies are appropriate and interpretable based on the characteristics of that agent. And then example activities can include PK studies to determine therapeutic agent levels, studies to confirm target access and target engagement, or studies to confirm a therapeutic agent's mechanism of action, pharmacodynamic and or in vivo efficacy studies, as well as assessment of potential off-target activities, addiction potential, or other potential liabilities such as preliminary safety. Now, I also want to point out, in contrast with past funding opportunities, addiction liability testing is not required for this program, although it should be addressed within the application, and I'll come back to that in a couple minutes. So the past two slides should hopefully give you a flavor of the types of activities this RFA may support. And again, additional examples are listed in the funding opportunity. This is not an exhaustive list. That being said, there are activities that this RFA is not intended to support. Next slide. Okay, so non-responsive activities include projects focused on target identification and validation, basic research or studies of disease mechanism, animal model development, opioid sparing projects or projects targeting the mu opioid receptor, projects consisting entirely of computational studies. So this would be virtual or in silico screening and characterization without any follow-on in vitro or in chemical testing. And then also projects in the optimization or IND enabling stages, which includes projects that incorporate a hit to lead or structure activity relationship driven chemistry campaign to identify agents with activity against a target of interest combined with desired drug-like properties. And then lead optimization to improve efficacy or pharmacokinetics, as well as the IND enabling studies. Now, for this last point, it's important to note that all of these activities are covered by the subsequent funding opportunity for the Pain Therapeutics Development Program. So it's within that program you'll be able to do exploratory hit to lead work, conducting more thorough in vitro ADME and PK work, as well as more iterative chemistry campaigns that go into improving various properties of your asset. So I would strongly recommend that groups take a look at that funding opportunity and talk to us beforehand when trying to determine the best fit for your projects, because applications that are considered non-responsive will be withdrawn and not reviewed. Next slide. Just a couple more activities that are considered non-responsive would be manufacturing, discovery and development biomarkers, devices, drug device combinations, surgical procedures, that type of thing. And then clinical research, which is defined by NIH as human subjects research, except for research that meets the exemption 4 criteria. And if you're not sure what that is, please reach out and we can provide additional information. And then finally, clinical trials. This is a clinical trial not allowed, NOFO. Next slide, please. Excuse me about that. All right, so now that I've gone through the types of activities supported, let's go back and put them into the context of the funding mechanism. As you might be thinking, well, what should go in the R61 versus the R33 phase? And the answer, of course, is it depends. So applicants should propose R61 and R33 activities based on the stage of the project relative to identification of potential therapeutic agents and what is needed to inform downstream activities or further de-risk the project. And then another way of putting that is the R33 work should be dependent on the results of the R61 to some extent. So just a couple of examples of how this might work. You could do initial development and validation of the assays in the R61 phase with the screening and hit confirmation or characterization in the R33 phase. Some groups would do assay optimization for increased throughput and potentially a pilot screen in the R61 with then a full-blown high throughput screen in the R33 phase. Others might do small scale screening, synthesis, or bio and physical chemical characterization of potential agents in the R61, followed by more extensive cell-based functional and electrophysiology-based assays in the R33 phase. And then for applications proposing in vivo studies, the R61 would most likely include some ADME and PK that would inform dosing, followed by the in vivo studies in the R33 phase. Again, these are just a few examples. It's up to the applicants to decide what they think should go in each phase, as well as the timing of each phase based on what makes the most sense for the science and for the project. Now, applicants typically request 1 versus 2 years for either of the phases, although 1.5 each is possible. The key is that 1, the total can't be more than 3 years, and 2, they can't overlap in time. Rather, the R61 and R33 phases must be separated by the go, no go decision point that's gated by milestones. Next slide. Okay, so what do we mean by milestones? For the purposes of this NOFO, milestones are quantitative goals used for measuring success that support go, no-go decision making. So milestones must be proposed in the application and clearly specified. If we can't clearly see that the application includes milestones, it's going to get withdrawn. Milestones should be well-defined, measurable, and include quantitative criteria for success that are scientifically justified. Okay, so specific aims or a list of activities are not milestones. Rather, they should have quantitative metrics that are results driven. So for example, while a specific aim would be to optimize and validate an assay, which is more of an activity, the milestone should be more to clearly define criteria such that someone else could objectively evaluate whether that assay has indeed been optimized and validated as intended. So really consider what criteria are needed, whether it's a Z prime score, acceptable signal to noise, or coefficient of variation, should compounds show a dose response or a needed fold change compared to controls, that kind of thing. Another type of milestone we often see is something where X needs to show a statistically significant difference compared to Y, which is in a way quantitative. But what does that really tell you in terms of how robust the result is? Rather, a much stronger milestone would include an effect size and clearly define the comparators, specifically including both positive and negative controls, number of animals or replicates, that kind of thing. So we would encourage you to consider, what do you really need to see coming out of the activities to warrant continued investment in this space? Now, milestones will be assessed as part of peer review and should factor into your scores. But prior to award, NIH program staff will work with the applicant to finalize milestones based on review recommendation, as well as the goals of the project and the program. And then, as mentioned previously, milestone evaluation to transition to the R3 phase will be done by administrative review. Next slide, please. Okay. Notable elements of an application, so things that need to be included or addressed that are more specific to this RFA. The first thing being a required other attachment or for an intellectual property strategy or IP strategy. So this is not extensive. It's a maximum of 2 pages. There's definitely no expectation that you should have patents filed at this stage, although if you do, then that's something you want to note. But it is something that you need to consider and have a plan for. So this section is meant to convince reviewers in NIH program that this has a path forward and that there's no existing IP that blocks you from doing what you want to do in this program, but also show that you're thinking about those next steps and what the competitive landscape might be. The last thing you want is for reviewers to question whether there's a path forward, even if the work proposed in the 3-year grant is successful. Now, additional details for what to include in this section are in the application instructions, and we would strongly encourage you to prepare this section in consultation with your institution's tech transfer office. So you really want to start that conversation early. Next is for the research strategy, which includes instructions for 5 subsections: significance, approach, innovation, therapeutic discovery plan, and then the milestones and timeline. Notably, for the approach section, you need to include activities for both the R61 and the R33 phases. So everything is laid out from the beginning and assessed by review. In terms of the therapeutic discovery plan, here is where you are going to briefly describe those next steps and how this work will significantly advance the project towards the Pain Therapeutics Development Program or other later stage therapeutic development opportunities. So again, we would strongly encourage you to review the PDDB entry criteria and that funding opportunity when preparing this section. So you want to convince reviewers that the proposed experiments are part of a well thought out and clearly defined therapeutic discovery plan. So we're looking for something a little bit more than after we're done here, we're going to apply for PDDP. Okay, and then we've already talked about the milestones, but there should also be an associated timeline for those project activities. And this RFA is also subject to NIH's data management and sharing policies, but it's important to note that HEAL has some additional specifications on top of those standard instructions, which notably includes a list of HEAL compliant repositories that you would need to choose from. But HEAL also offers a lot of resources to help applicants prepare this section. And these are outlined within the NOFO, but I would also encourage folks to check out the resources provided online, which investigators have found to be really helpful. So I've included a link for that here. And then finally note that the elements in the slide that are underlined represent required elements. And if your application does not include any of these, it will be considered incomplete and withdrawn prior to review, regardless of how amazing the rest of the application might be. So please pay close attention to those. Next slide. Okay, so things to consider. I'm highlighting a few elements here, but again, a lot of this is more explicitly laid out in the application instructions and might vary based on what you're proposing to do. So please read through those carefully. Okay, potential for addiction liability of either the target or the therapeutic asset. This needs to be addressed in the application. Again, experiments to explicitly test this are not required, but it should at least be addressed. So for example, in the significance section, you would include rationale supporting the limited addiction potential for the proposed target or therapeutic approach. That being said, based on your target, it may be something you do want to explicitly test early on, and that's fair game for reviewers to comment on as well. Okay, so for assay development and screening, consider, is the assay scientifically sound, or does it make sense based on the target and its biology? Is the library appropriate for your needs? And what defines a hit in the primary screen? What kind of throughput is needed? Are secondary assays also needed for orthogonal or counter screening? So consider the role the assays will play in the therapeutic discovery and decision-making process, and how potential assets might advance through a testing funnel. So a flow diagram can be really helpful here. Now for in vivo work, really consider whether the asset has been sufficiently characterized. So again, applications proposing in vivo studies must clearly identify and describe the therapeutic agents to be tested and provide evidence such that reviewers can evaluate whether the properties of that agent are suitable and appropriate to justify in vivo efficacy testing. You want something that's been characterized enough for a meaningful initial preclinical proof of concept study. You should also provide strong rationale for the chosen models and the outcome measures that you'll be using. And this includes rationale for what would be considered a clinically meaningful effect size. Also discuss elements of rigor and transparency as it relates to the specifics of your project. So we're not looking for blanket statements that you'll be sure to use appropriate controls and do a power analysis, okay? State what the controls will be and consider using both positive and negative controls. What are potential biases associated with the specific methods that you'll be using, and how are you planning to account for that in your analyses, that type of thing. Also worth highlighting here is that translation requires collaboration and a multidisciplinary approach. So in addition to the relevant pain biology expertise, we would strongly encourage you to form collaborations with individuals with expertise in various aspects of drug development, and those familiar with what the desired end product should look like. So technical experts with experience developing drugs of that particular modality, as well as biostatisticians, clinicians, maybe that type of thing. Next slide. All right, I wanted to touch on a couple points about the review of these applications. So that will be done in a special emphasis panel convened by NINDS review branch. All the applications will be reviewed together. The review panel will also be multidisciplinary and very translationally focused. So you'll have reviewers who know the pain biology side of things, or potentially the relevant expertise for a specific condition that may be the focus of your application. But you also have reviewers who are experts in therapy development. Okay, the review is likely to include researchers from both academic and industry backgrounds, and potentially clinicians as well. So you're gonna have a lot of different perspectives looking at your application. So just know that this is your audience. Now, overall, review will be assessing the likelihood that the work will significantly advance projects towards PDDP. A part of that may be assessing whether the target is really ready for prime time. Has it been sufficiently validated? Is the team multidisciplinary? Does it have all the needed expertise to conduct the project and plan for the next steps? Again, milestones factor into the priority score, whereas the intellectual property strategy does not factor into the score. So it's an additional review consideration. That said, reviewers are still going to look at that section and comment if there are any concerns. Next slide. Okay, just a couple more slides that should hopefully serve as a summary for the presentation. In terms of considering what makes for our competitive applications are going to be those that have a strong biological rationale supporting the intended target and approach, They'll include a rigorous study design and supporting data. We cannot stress this enough for translational research, the importance of the robustness, reproducibility, and transparency of experimental results. Quantitative milestones that allow for go-no-go decision making. Having a multidisciplinary team, note the multidisciplinary review that I just discussed. And the best way to write an application that appeals to such a diverse audience is to have a multidisciplinary team yourself who's helping you write it with all those different perspectives in mind. The increasing diversity of your team, as well as incorporating people with lived experience, would also be considered strengths for an application. And finally, having a well thought out discovery plan and discuss your intellectual property. So again, a therapeutic discovery plan, the team, the IP, all gets at the question of, does this have a path forward? So you want to show that you're thinking not just about the next few years, but the feasibility of moving this forward beyond this grant to PDDP or other later stage programs and ultimately the clinic. Next slide. Okay, so finally, just some more general tips. Carefully read the funding opportunity. Again, there's a lot more detailed information than what I've had time to go through today. Clearly mark which activities are in the R61 phase versus the R33 phase and include a timeline. Reminder, R61 and R33 phases cannot overlap in time. Make sure you include quantitative go-no-go milestones. Discuss rigor, both the preliminary data and the proposed experiments, and then pay attention to those non-responsive activities that I mentioned earlier and contact us prior to submitting an application. Send us a specific aims page to be, especially if you're not sure, but do it regardless, even if you do think you're sure. And then again, be sure to review the HEAL data sharing policies and available resources. Next slide. This is just a list of the contacts, so you'll have that later when you get a copy of the slides, my contact information, as well as all the different representatives from the different ICs that are signed on to the RFA. I've also included Dr. Pellimonter's contact information. She's the lead for the PDDP program, as well as Dr. Michael Shinsky for target discovery and validation or more general questions about the HEAL Initiative's preclinical and translational programs. Next slide. All right, so with that, we should have time for some questions. That being said, for project-specific questions, please contact us by email and include a draft aims page. Again, devil's often in the details, so having the draft aims page or an outline of what you want to do is really the best way for us to both to get the most out of such a call. Okay, and I think that's all I have. Probably you can stop sharing, Rachel. Okay, so let's look at what questions we have. Okay, can you break each phase into 18 months or some other allocation? For example, total program is still 3 years. Yes, so you can, a lot of people do 1 versus 2, 2 versus 1, but you can do 1.5 and 1.5. You can't have both the R61 and R33 awarded in the same fiscal year, which makes something other than that 1.5, 1.5 very difficult, if not impossible. Okay, second question. R61 is the first phase and the R33 is contingent upon meeting a specified go, no go branch point. How much detail in the research plan, oh, we moved around, is required when describing the R33 activities? All of it. So you want to, in your research strategy, you're including all of the activities for both the R61 and the R33. So reviewers are going to evaluate your entire plan for the 3 years. Is it acceptable for one PI to submit 2 proposals? Yes, so long as they're not overlapping and duplicative. That's standard NIH considerations for overlap. R61, R33 work should include PDPK. Depends what you're doing. So a lot of this is going to be earlier on when people don't have their assets identified, in which case we would not recommend PK/PD work. If you have something that's already been identified and well characterized, then you can include PK/PD work. So it really depends on where you are in this stage. So that sounds like something we should have a conversation about. Okay, yes, you will get a copy of the slides as well as a link to a copy of the recording of the webinar. Can we use a reformulation of an existing analgesic that's anti-inflammatory? So this one, potentially yes, potentially no. This is an example of something where you would want to reach out and talk to us to see what the specifics are and what you're planning to do compared to what's already out there in terms of the existing drug. Preliminary data is required. It can be from literature-based evidence. It doesn't necessarily need to be in from the lab, but if you don't have any preliminary data from your own lab, that is probably going to not do well in review. So you want to at least have and show reviewers you have confidence in the feasibility of the project itself, the type of activities that you're doing, that kind of thing. And you also need to have strong support for the biological rationale of the target, which again could come from work that's been published or work in your lab. If members of the multidisciplinary team are based outside of the United States, can work in their lab be included in the grant and funds used to support that work? So yes, foreign components are eligible and allowed as a part of this funding opportunity, but foreign applications, so applications directly from that foreign institution, are not allowed for this application, for this NOFO. Can a research associate apply funding as the principal investigator? Yes, I would imagine so. So the eligibility criteria for who can be considered a principal investigator is pretty standard NIH language, but that's in and spelled out in the funding opportunity. We'll give it a couple more minutes. See if additional questions roll in. Humanized mice are not feasible. What should we do? So I mean, in general, that depends on what you're trying to do. And if you're trying to do an efficacy study, then if there are more complex in vitro model systems that can be used in their place, that is a potential way to go about it. For in vivo studies, again, I would really encourage you to consider whether or not what you have in hand has been sufficiently characterized in vitro to really warrant the in vivo testing, especially if it's going to be something that's other than a rodent. I should also note that in vivo efficacy studies and that proof of principle data is not actually required as an entry criteria for the Pain Therapeutics Development Program. So it's really going to depend on the asset and the target and what's already known about that in the community, in the literature, that kind of thing. So I don't want everyone to expect that they need to have a very strong data package, including all these different animal models of efficacy data before applying for PDDP. Some of that might be needed in order to be competitive in PDDP, but that should really be a conversation that we have either between myself or Dr. Pellimander and her team. We can talk about the details, but I hope that answers your question. But if not, we can set up a call and discuss further. Will an application on cancer-associated pain be considered in this RFA? Yes. So the type of pain does not matter. This is the HEAL initiative, so it covers all types of pain within the NIH mission space, so all the different ICs. So NINDS will hold all of the grants, so they'll all be assigned an NINDS grant number, but all of the other IC reps are involved in terms of developing pay plans and milestone evaluation and that type of thing. Give it more time in case some late breaking questions come to you. I hope I didn't miss anyone's question. All right. Sounds like questions are done from now, but you have my contact information hopefully, and if not, we'll be sending out a copy of the slides and the recording to everyone that registered for the webinar. So please feel free to follow up with an email and an Ames page if you have it would be great. If you don't, even just a brief outline of what you're proposing to do so I can roughly make sure you're in the right place and talk to the best people, that would be ideal. And then thank you everyone for your attention and time out of your day. Rachel, are you going to stop the recording or do I need to?
I don't think I can.
All right, then I'm going to hit the end button.